A-Level Biology 9700, Paper 4, Variant 2, February, March 2026. And in this video, we will discuss question number 5 and 6. So the question number 5 is, question number 5 is the sarcomere is composed of many structures and is the functional unit in the contraction of striated muscle. So complete table 5.1 to match each description with the correct region of the sarcomere. So they have given us the regions of sarcomere, but they and which we have to fill, and description has been given there. That is, we have to find out the region where the only myosin present, where the actin and myosin present, only actin present, attachment for myosin and attachment for actin. For this purpose, we have to go the parts of the sarcomere where all these are present. So the first was the where the only myosin present. Here you can see the thick filaments are of myosin and the thin filaments are of actin. And you can see the thick filament. The first to fill in the blank which we have to fill, that is where the only myosin is present. You can see the only myosin is present, which is of thick bands. It is present at H zone. So they have highlighted the zone, you can see. So this is the H zone where the present. Then actin and myosin present, both are present. So both are present at the, you can see actin is present and myosin both are present. So this is the A band. And the only actin present, only actin is present at a place, you can see that this is the place. So this is the I band. And then attachment for myosin. Attachment for myosin is there. This is the, or this one. So this is the M line. And then attachment for actin. Attachment for actin. You can see this is the Z disc. So let's move the space where we have to write all these. As we have only myosin is present in H zone. And actin and myosin both are present at A band. And only actin is present, that is I band. And attachment for myosin was the M line. And attachment for actin was Z line. Next is part B, and part B is hypocalcemia. Wait. Hypocalcemia is a condition where the blood calcium ion concentration is below the normal range. Explain the effect of the blood calcium ion concentration on the functioning of the sarcomeres of striated muscle fibers. So we have to write down the effect of the low blood calcium ions concentration on the functioning of the sarcomere of striated muscle fibers. Students know if they are there are low blood calcium ions concentration, then what can happen? Then first of all, the less calcium ions will enter the sarcoplasm. So when they will be low, then there will be the reduced binding to troponin. And when there will be the less binding of the troponin, so myosin does not move. If it will not boost, then there will be not the formation of the cross bridges cannot be formed. So these are the effects when there will be the low blood calcium ion concentration. Let me write on the given space. Here, fewer calcium ions are released from the sarcoplasmic reticulum into the sarcoplasm. Then a nerve impulse so fewer calcium ions bind to troponin. So the troponin changes shape or not. Tropomyosin fails to shift away the myosin binding site. On the actin filament. On the actin filaments, leaving them blocked. Myosin heads cannot attach to actin to form cross bridges. Preventing the power stop. And stopping the muscle contraction. So it was question number 5. Moving towards the question number 6, and question number 6 is protein production in a prokaryote can be genetically controlled. One example of the control of protein production in a prokaryote is the lack operon. The lag operon is functional when glucose is absent and the lactose is present. Complete table 6.1, to which shows the information about the four genes associated with the lag operon. So here you know that they have given LAC-I, which is the type of, we have to tell the type of the gene. So name of the protein which is produced, that is the repressor protein. So the function of protein produces the binds to operator to prevent RNA polymerase from binding to the promoter. So it means the type of the gene which will do this, that is the regulatory genes. That is regulatory gene. Then the second row is the lag G structural beta. The name of the protein produced is beta galactosidase. So the function of the protein is it hydrolyzes the lactose into glucose and galactose. And now the third row is type of the gene is structural lactose permease, and the function of the protein produced is the membrane transport protein to increase cellular uptake of lactose. So it is the name of the gene is, because it is again structural type. So this is the LAC1. So the last row they have completed by themselves, that is the LAC-A is of structural type genes. So trans-acetylase. So acetylase galactoside sugar residues to detoxify the cell. So it was all about this part of the question. Now moving towards the B part of this question, that is insulin can be produced as a recombinant human protein using genetically engineered Escherichia coli bacteria. Recombinant insulin can be used to treat type 1 diabetes. Describe the advantages of using recombinant insulin instead of insulin from animal source to treat type 1 diabetes. So here we have to write the answer about the advantages of using recombinant human insulin. So there are so many different advantages of using this recombinant insulin. That is, first of all, the structure, its structure is identical to the structure of the human insulin. Secondly, it is more effective and fast acting as compared to the animal insulin. Uh, Thirdly, no immune response or lower risk of allergic reactions by using this. And reliable and large scale production can be done with this. Uh, then it has low risk of disease transmission. So it eliminates the risk of transferring animal pathogens or infection to humans. So ethically and religiously it is acceptable. And then it is cost effective, that is the production processes using bacteria fermentation can be cheaper into the long run compared to the extracting and purifying insulin from animal pancreas. So these are the different factors which we can write right over here. First of all, recombinant insulin has the exact amino acid sequence. Sequence as human insulin. More effective than animal insulin, less likely to trigger an immune response than can be produced in unlimited supply to meet the demand. Demand. Eliminates the risk of transferring animal pathogens. Animal pathogens to humans. Cost effective production processes using bacterial fermentation process using bacterial fermentation can be cheaper ethically and religiously acceptable. So it was all about the question number 5 and 6. Please like, share, and subscribe my channel. And because in this way you will be able to watch more videos. Thanks for watching.